Hello everyone, my name is Benjamin, and today I speak on behalf of my colleague. There might be a link between early bladder infection and onset of interstitial cystitis. One evidence of that is that about 50% of our patients have medical histories of bladder infection, with E. coli being the prime infectious agent. E. coli is a bacteria that has a lipopolysaccharide layer that binds the toll-like receptor 4 of many cells. And toll-like receptor 4 activate pro-inflammatory then apoptotic response by recruiting a lot of factors such as Tract6. And Tract6 can, on its own, activate NF-kappa-B and JNK pathways. Another evidence is that during sepsis, a lot of tissue accumulate proNGF factors, which is a factor that binds the P75 receptors, and the interaction of the proNGF factor with its receptor P75 is known to generate inflammation and apoptosis. What is particular about these factors is that the proNGF also accumulates in bladder tissues of patients with interstitial cystitis. And there was a very recent article showing that blocking the P75, so basically blocking the interaction of proNGF with P75, can attenuate inflammation in the brain of septic rat. So this basically suggests that there might be a crosstalk or an interplay between the toll-like receptor's annual transduction and P75 inflammatory axis. To answer a research question, we put through urothelial and small cell culture out of bladder from Sprague belly rat. And all of these cultures were then treated or untreated with the THXB molecule, which is the P75 antagonist. Then these cells were subsequently either exposed to LPS or not, making four groups which were subsequently all evaluated for protein expression of inflammatory signaling pathways or association of the Tract6 factors to P75. So to start with, we found that urothelial cells exposed to LPS increased the cytokinetics of TNF-alpha and its release in cell medium. And that when we pretreated these cells with THXB, there was subsequently a very significant decrease in both form of TNF-alpha, suggesting that the P75 antagonist might interfere with TNF-alpha cytokinetics and its release in cell medium of urothelial cells. Next, we found that urothelial cells exposed to LPS alone increased partially Tract6 association to the P75 receptors, as well as ERK signaling pathways activation. And adding the THXB molecule did not have any effect on these pathways, suggesting that P75 antagonists can maintain activation of Tract6 and ERK activities only in urothelial cell. Finally, small muscle cell exposed to LPS show to increase Tract6 activation, as well as NF-kappa-B and JNK inflammatory pathways, and subsequently adding the THXB molecule decreased significantly all of these pathways, which suggests that the P75 antagonist can block LPS-induced activation of Tract6, NF-kappa-B, and JNK. So in light of our data, we suggest that there is cell-specific response to the P75 inflammatory axis in bladder cell, and that the toll-like receptors can interplay with P75 via the Tract6 factors in the bladders, and also that blocking P75 can attenuate inflammation in bladder cell, although we understand that more investigations are needed to validate this anti-inflammatory effect in animal model.